billions of years. Oh, let me turn this on. This story begins billions of years ago when a simple single-celled bacteria, cyanobacteria, figured out something very clever, how to harness energy from our environment. And in the process generated oxygen, a waste gas. And over time, this gas saturated its sinks and began to fill our atmosphere. So today, our atmosphere has 20% oxygen by volume. We all rely on oxygen. It shouldn't be in the atmosphere without life, without plants and photosynthetic bacteria, because it's just highly reactive. So if there's an intelligent civilization orbiting a planet, on a planet orbiting a nearby star, and they have the kind of telescopes we're hoping to build, and they look at Earth and see oxygen, they will be highly suspicious because it has to be continually replenished, and they might suspect there's life here. They're more likely fighting over data analysis, attribution, false positives. But this has been the hope and dream for exoplanet astronomy. And if you dig back, actually, if we dig back, the first reference I've ever seen to oxygen as a sign of life on another world, not an exoplanet, but a solar system planet, comes from Cambridge, actually, from genes, as a footnote in this textbook. So the reason why there's so much hype around the search for signs of life by way of biosignature gases is with the Webb telescope and the amazing capability for transits. Now, I know you all know what a transit is, but I just wanted to give a quick review and show you a really cool data that you might not have seen yet. So here's the transit light curve. You've all seen this before. It's showing you relative brightness in percent as a function of time. There's this characteristic drop. That's the planet area to the star area. Now, if you look at the cartoon at the top, what do you see? You see a fake atmosphere in gray. So I want you to now think of this like the planet, the dark part of the disk, takes out a certain fraction of the starlight. And at wavelengths where the atmosphere is fully transparent, that's what you get. But at wavelengths where the atmosphere is very strongly absorbing, the planet looks a tiny bit bigger. Are you with me? So we're going from like that dark circle to like that combined annulus and dark circle. So the whole thing of the transit transmission spectroscopy is we're looking for a change in transit depth as a function of wavelength. So here is a hot Jupiter with the James Webb Space Telescope. It's WASP 39b. And what you're seeing is the transit light curve. You're seeing in black, that's the best fit to all this data. W's wavelength in microns. And your job right now is to look and see which wavelength the points drop below this best fit. Where's the transit a tiny bit bigger? Where the atmosphere is strongly absorbing. So do you see a little drop somewhere? And just shout out what wavelength it's at. Wow. You have seen evidence for an exoplanet atmosphere by eye. That's so amazing for our field. We're nowhere near high precision cosmology, of course, but we are just so proud of where we're at today. And here you're seeing the transit depth in percent as a function of wavelength in microns, and that's what you saw, that around 4-ish microns, the planet looks bigger. This is, we call 35 sigma detection. You can see it from across the room. And that hasn't been true in all the exoplanet atmospheres talks you saw before Webb launch. So in this case, I'm just showing this to you to impress upon you how we're in this great time with transmission spectroscopy. That's due to carbon dioxide, by the way, just a tiny amount in this hot giant planet atmosphere. It's, like a powerful greenhouse gas. It's a very strong absorber. And at the bottom, it's just showing you some cross sections. So by eye, you can see, convince yourself that it's carbon dioxide. So the point of this is just that with transmission spectroscopy, the community aims to push down to smaller planets that are the right temperature for life, not this big hot Jupiter with a puffy atmosphere. That's what I'm going to be, that's kind of the basis of the excitement, that for the first time, we astronomers, we humans, we have the first chance to find signs of life by these biosignature gases. I left a few things in here for your notes because I don't have time to talk about it. We heard about some of these earlier in the week, and your resident expert, Madhu, can answer any questions anytime. So it turns out that our Earth only had significant oxygen at this 20% or even a bit lower for 1 billion years. For you, that's not a long time for extragalactic cosmology. But if we've been around for 4.5, 5 billion years, that's just some fraction of the time. So we need to ask ourselves what other gases might be biosignature gases. Well, I found this super interesting factoid that it's not just oxygen made by life, but it turns out that with the exception of the noble gases, any gas in our atmosphere to the part per trillion level, PPT, actually is made by life, although it usually has a dominant source that's not life. So I could challenge you to think of a molecule, and honestly, most molecules you could think of as a physicist, they actually are made by life. And it's just incredible, like benzene, even ozone is produced inside some cells. So I embarked on this kind of decade or 2 decade long journey to think about every single molecule that is in gas phase at habitable planet conditions. Because it turns out that our life is producing thousands of molecules, but some have very obscure reasons, signaling or hormonal. And on another planet, it could be some other molecule being used. So we wanted to tabulate all these molecules so that eventually we could be prepared to recognize biosignature gases of any kind. So this is kind of a separate topic. And you know how sometimes in astronomy, we had the question, when do you stop? Why are you doing what you're doing? And sometimes you don't know. You're just sort of following something you feel like is right. And this turned out to be a very rich field for a lot of reasons. We just call it chemoinformatics. But the point that might be more relevant for you was I put a student would have like a few different gases or classes of gases, and we kind of put it through this machinery, triage, where we get a spectrum, either from, not our lab, but that existed from laboratory data or sometimes from theory. Most of the molecules don't have spectra, by the way. And we need to ask ourselves, is the molecule distinguishable from background gases in the atmosphere? Is it distinguishable compared to other potential biosignature gases? If yes, we go onto the right side and we evaluate the gas as a potential biosignature. You know, largely it's about chemistry. And you might have seen from some of the talks earlier in the week that, and at least for if you want to follow or understand exoplanet atmospheres, it's turning into a field of astrochemistry now. And that's true for biosignature gases because they're produced by life, they get destroyed. And if they're just kind of destroyed so rapidly and immediately, they're not suitable biosignature gases. But we've gone through a lot of these, and I'll touch on that a bit later. But the point that becomes relevant later in the talk is about photochemistry. We ask ourselves in our models, what kind of spectral feature do we need of a gas, to detect? And then the key is how much has to be produced. Because if you find that you need, because the gas is destroyed, so it's a matter of comparing sources and sinks. Okay, so if you end up finding that you need an outrageous production rate that, say, billions of times more than oxygen produced on Earth, it's just not feasible. You have to have some reality check. And so this whole photochemistry is really key to everything. So that was kind of my background. And where we're at now, we have rocky exoplanets in the habitable zone of host stars. We have established methods to observe exoplanet atmospheres. It's not just transmission spectroscopy, but for the future, it's direct imaging and high spectral resolution cross-correlation. I obviously just touched on it very briefly, but I left some references for your notes. We, that's myself and many others, have a list of and a framework for how to predict and how to possibly identify and interpret biosignature gases. So it seems like we're in a good place. And out of all those gases my team has looked at, and we certainly didn't need this overly complex machinery to study it, but I actually do have a favorite gas out of it that popped out right away of being incredibly valuable as a biosignature gas, and that gas is phosphine. Because phosphorus is an essential element for all of our life on Earth. But on Earth, we have really little hydrogen compared to oxygen. So phosphorus is present here as phosphate. It's not a gas, but just phosphine gas is extremely rare on Earth. It's highly thermodynamically disfavored. We have very little hydrogen lying around. And on Earth, it is literally only associated with life. We make it as a toxin, as like a pesticide. Life in oxygen-free environments makes it. So I love this gas because it kind of checks all our boxes. And we have 3 boxes when we think of a biosignature gas. One is no false positives. It's really never strictly true, by the way, but kind of on the whole, bigger picture level. In terms of photochemistry, if we have an atmosphere without oxygen, phosphine can accumulate with relatively reasonable production rates. It doesn't just instantly get destroyed. And also, it has somewhat unique spectral features depending on what the background dominant atmospheric gases are. And on the right, I just wanted to show you these cross sections. in cross sections as a function of wavelength and microns. If you look at the white curve, that's phosphine cross section at room temperature. And you can kind of just glance at the other colors to see, does phosphine have any unique spectral features? You can see in the top left, for example, it pokes out above the water vapor feature. So on the whole, I would have said this was my #1 absolute favorite biosignature gas. And my team had worked on this for a few years. We'd written a few papers, sort of reviewing all the ecology on Earth, making a prediction paper, hashing it out for exoplanets. When we, oddly enough, heard of another astronomer across the ocean, Professor Jane Greaves, also working on phosphine. And she went out on a limb and wanted to find signs of phosphine on Venus. I think some of you have heard about this. And okay, you've all heard about it. And for me, this was like a I don't know, Back to the Future kind of thing about exoplanet biosignature gas in the future. I feel like I've kind of lived through discovering a biosignature gas, and it's not pretty. So we have these 3 questions now that we did have all along, right? Is the signal real? Is the molecule correctly identified? And are there other ways to make this molecule that's not from life? Well, the controversy surrounds mostly the first point, because we have data can be, that's another talk. That's for you to invite Jane Greaves to come here and hash that out. But let's just say large majority of people have not recovered the signal in the ALMA data. We also have JCMT data, and people have recovered the signal. There, people want to attribute the line, it's a 1 millimeter line, not to phosphine, but to another molecule, sulfur dioxide. You know how out in the millimeter and centimeter rotational lines, they're very well spread out. It's not like a forest of lines we get for rotational vibrational lines in the near infrared. And so it turns out that this line of phosphine, it just overlaps with a line of sulfur dioxide, which is really definitively on Venus. It comes out of volcanoes. I don't mean to hash it all out, that wasn't my intention, but we don't think it is because you'd have to have a lot of sulfur dioxide. It's a higher level transition, and we don't see that in near simultaneous data. But as astronomers, we know that for Venus or for an exoplanet, we could get those first 2, right? We could get more data. We can have higher spectral resolution with new instruments. That's not the problem that I see with this whole story. The question is, can we attribute a gas to life or not? And this really is going to take a long time to hash out. You mostly heard Lisa Kaltenegger's talk about some volcanic things. I think closer to our memories is Ray's talk from yesterday. Do you remember all that detailed chemistry? Hopefully you were paying attention. It's very detailed. But the problem is we have these planets with a lot of unknowns. Think about not just no initial conditions, but imagine you're trying to solve a problem with no boundary conditions, or your boundary conditions are so vast and diverse from each other that it's turning into like a ginormous thing of free parameters. And that is where we're at. I have a couple of examples for you, but in this cartoon, it's just showing you geoflux and bioflux mixed together, get photochemically altered along with atmospheric escape. And some molecules, they get rained out, they get absorbed by the surface. So you could imagine we could have our own version of the lustrous TNG and work every last thing out. But our issue is that we don't have hope for making the small-scale observations. We don't have anything that's equivalent to particle accelerators or weak lensing or some way to probe what's really going on. I have one example I'll give you. It's sort of related. It's about our planet Venus, where it's a massive carbon dioxide greenhouse atmosphere. But if you were to make remember I told you chemistry is everything? If you make the photochemistry model, carbon dioxide gets split up by ultraviolet radiation, and it recombines in almost equal parts to carbon dioxide, carbon monoxide, and oxygen. So if you're just doing your model as an astronomer or chemist, that's what you would expect. Venus has 97% carbon dioxide. What's going on is there's a catalyst present, a molecule that makes the reaction preferentially go back to CO2 while itself remaining unchanged. And people predicted this was a kind of chlorine species in the 80s. And they've kind of identified it, but it doesn't seem to have been totally solved yet. So there's this insidious photochemistry makes it very complicated. So there are a long list of molecules. I'll probably leave this for your notes, but just so you know that people are really working on this in all the contexts of all the molecules. But that question is out there. Can we ever claim a robust biosignature gas discovery? I think it will take us the next 50 years to answer that. But let's just summarize that problem we have right now. Thousands of gases are produced by life on Earth. The problem is that we have the context for false positives will remain unknown, even if we can robustly all agree that we have identified a specific gas. So what's our solution? I'm going to let you mull that over for a minute while I tell you what I've been doing while mulling this over. So it actually turned out that on Venus, I forgot to mention, it wasn't just, on Venus, by the way, Pioneer Venus, a NASA probe that went to Venus 4 decades ago, did also have some evidence of phosphine. But what have I been doing? Well, it started in the pandemic summer when this phosphine, we were wrapping up with Jane Greaves, the whole phosphine thing. And it was, I forgot to mention one other key thing as well in the interpretation, that we wrote like 100 pages and went through all the chemistry, everything that's known about phosphine on Venus. It's not all known, but lightning, volcanoes, meteorite delivery, and none of those could produce phosphine in the amounts required based on what we know, but it was still really highly challenged. So during the pandemic summer, this was all unfolding. And if you remember, there were, if any of you had a teenager, I had a 17-year-old, there's no jobs, nothing to do for these kids at all. So I put my 17-year-old son at the time onto the Venus project. It was like an internet project. He finished it in a few days, honestly, and then he pushed to do real experiments. So it turns out that we decided to set up like an outdoor lab. You cannot order a lot of sulfuric acid to your house. Sulfuric acid is a very, very nasty chemical, and it's what the Venus clouds are made of. And it actually, by the way, it turns out that Carl Sagan was the first one to propose there might be life in the clouds of Venus. The surface is too hot for life of any kind. But just like on Earth, if you go in an airplane or climb a mountain, it gets colder and colder. And those clouds, they're at the layer 50 kilometers above the surface. They're just the right temperature for life. But we think he didn't know they were made of sulfuric acid. People didn't know at the time. And acid, it's so, so nasty. So I just want you to pose it to you. You're a 17-year-old. It's a pandemic summer. There's literally nothing to do. It's an outdoor lab. It's like today, green. You're looking all around. What's the first thing you're going to put in sulfuric acid? Outside. You got an outdoor lab. You're just going to try out some experiments, putting things in sulfuric acid and see what resists or what gets destroyed.
Sugar.
Sugar. Okay, sugar is bad. Okay, you're pretty nice, though. Uh-huh. Oh, no. Okay. Safety first, guys. That's what I tell my kids. Safety first. This stuff is, sulfuric acid will burn a hole in your clothes and your skin. Come on, you guys are friendly. What you're looking around? An insect. There you go. So before I could say, please don't do that, he took an ant and put it in sulfuric acid, and it had a seizure and turned to goo before you could say anything. It was really awful. And that ant, and later on, don't do that now. You can Google sugar in sulfuric acid. Our DNA has a sugar molecule. It will just convince you that no life at all could survive in sulfuric acid. Yet, what we have been doing, that's my team and an extended team, and same son is now finishing up biochemistry and can actually do something real. We actually have started to seriously consider sulfuric acid as a potential solvent for life. And I feel like I'm on the most incredible journey of my life. We put, this was his project, amino acids in sulfuric acid. And with one exception, they're all stable for a very long period of time, like a year. Half of them are chemically modified, but they are stable. I put a little figure there of NMR. Don't have time to explain that now. We also put our nucleic acid bases, so although our DNA is unstable, parts of our DNA are stable. These are getting protonated and chemically modified, but we're now working towards a DNA-like molecule, not our DNA, but swapping out parts, to show that there could be life in sulfuric acid. I'm not saying there is life on Venus, but there could be. And one more of these I just want to touch on. You can take what people are trying to do in the lab to create life in water, like to try to understand the origins of life. They take fatty acids with a polar head, put it in water, and if you put the right amount, they point their head outward to interact with water, and they make like a spherical vesicle, like a very super primitive type of cell membrane right size. Well, you can do this in sulfuric acid, and it also forms that same structure. In fact, more easily, because sulfuric acid is more polar. And in some of these things I've just mentioned, it's crazy, but there's hints of this or work been done decades past when people were just studying pure sulfuric acid without knowing anything about Venus. So I started out doing this to motivate missions to Venus, small focused missions where we can go to Venus and look for life directly, a subject too taboo for large government agencies. But we have our first largely privately funded mission to Venus targeted for 2025 with Rocket Lab. It's more of a tech demo. It's just a very quick mission, and please ask me more about this later. So I definitely have had to take like a mental break from the search for life because of the whole phosphine on Venus and all of this. So what are we going to do? This conference has been incredibly inspiring. I loved all the data, all the models, and my head is spinning right now because one option, one idea is that we live with Biosignature planet candidates. I have no doubt that people in this very room will be finding Biosignature gases, but they'll be candidates, right? We could imagine investing in large-scale computer models of planet interiors, mantle, atmosphere, oceans, but yet, like I said, knowing we'll never have a planetary version of these small-scale observations. Madhu was my first student, my first ever PhD student at MIT, and we introduced this thing, we called it atmosphere retrieval, a way to bound all the possibilities out there. And while you may be fine with that for atmospheric constituents, would you be fine with that if we want to tell you we found a sign of life? But here are all the possibilities it could be with some kind of probabilities. It's not very satisfying. So I do have 3 solutions for you, and you can mull these over. You can pick your favorite. The first one is to go after an Earth twin around a sun-like star, as opposed to like a super Earth around an M-dwarf star. And we have a lot of hope for this in the future. The ELT, we have space-based direct imaging. It may still have that unsatisfactory scenario, but maybe with something a little more familiar, like an Earth with oxygen and water, it might help. Another option that people have been entertaining are technosignature gases. Gases produced by artificial means, either accidentally or for a specific purpose. And remember all my molecules, all small molecules in this chemoinformatics, we are quantifying interesting things in life. Our life on Earth avoids fluorine. Fluorine is a highly electronegative atom. It's like the super glue of atoms. And there's no real reason for life anywhere to use fluorine when there are other functional counterparts it could use instead, right? If it's so hard to break up an atom off to carry out a chemical reaction, it won't happen. And people love these complex molecules that include fluorine as technosignature gases. Really, we could do number 1 and 2, and we will do those for sure. But in my opinion, what it's really going to take is something completely different. And this is where, if you're the under 30 crowd, you should really be paying attention. Because it is literally going to take a whole new paradigm for us to be able to get close, to do something on that version of the small-scale observations. We have ideas that people, let's say, love to hate. The Solar Gravitational Lens Telescope that would go 500 AU from Earth and would use our sun as a gravitational lens to get down to 10 square kilometers around a targeted planet. There's Starshot, an idea to send thousands of space chips with solar sails to zoom by a planet and snap some photos. But whatever our future is in the next 50 years, we know it's going to be an exciting one. Thank you for listening.